So we're going to go through the cell-mediated immune response first, and then we'll do humoral immunity. So we discussed the thymus, and so remember T lymphocytes, they're made in the bone marrow, then they're released from the bone marrow as little cute little thymocytes, and then they go to the thymus. And in the thymus, we learned there's sort of 2 rounds of screening. The first round is called positive selection. They need to be able to recognize the self barcode, so the major histocompatibility complex, because if they can't read that, then they're of no use. And so if they don't recognize that, then they go through apoptosis. Then they go to the next round, and that's negative selection. And we just learned that in that little cleft, that's not the right picture, but in that little cleft of the MHC, that's where a cell is going to just display either self-antigens or corrupt pathogen antigens. And what we don't want is a cell that is going to recognize our self-antigens, and so they'll go through apoptosis. The other thing that happens is when they leave, then they are going to get their job. So just like when you graduate, you get a job. And so when they leave the thymus, they will either get a CD4 marker or a CD8 marker. And these markers are things that scientists use. I used to work with lymphocytes. All lymphocytes look alike. And so I would have to sort out my cells based on this CD, so cell determinant marker or cluster differentiation marker. And CD4 cells are destined to be the generals of the army. They are going to be the talkers, so the T helper cells. And then if the cell leaves with a CD8 marker, that means it is now going to be an assassin. So it's a cytotoxic T cell. So they're going to leave the thymus, and they leave as naive T lymphocytes, and that is a lymphocyte that's just never seen battle before. So our 2 populations, we have our, and I'm sorry they get lots of different names. So we have our T helper cell, CD4 cell, T lymphocyte. That is going to be a cell that's just going to release cytokines. It's going to release chemical messengers for the immune system. And so it communicates and it coordinates both the adaptive branches of the immune response and also talks to some of our innate cells as well. These CD8 cells are going to be a lot like the natural killer cells, only they're going to be targeted. So they're going to be assassins. So they'll recognize one antigen alone. The CD4 cell, like I said, its job is to release cytokines, and so it's a talker. So we learned about the endocrine system. Those are all communication signals. These are just communication signals between the immune system. Okay, so cytokines, interleukins is a type of that. So the T helper cell is the most pivotal cell in the immune system. It is the one that's going to be taken out by HIV, so the virus that causes AIDS. But the CD4 cell, T helper cell, is the talker. And those signals will then activate our assassins, so the CD8 cells, the cytotoxic T cells. So from the thymus, like I said, they're going to leave with their marker, their job, but they are naive. Where they're going to go, they are going to take up root in the lymphatic tissues, and we looked at our histology on Monday, and we saw a whole lot of lymphocytes. T lymphocytes will do more moving around the body than B lymphocytes, just because their mechanism, what they have to do is they have to crawl over all the cells and see if they're okay. Okay, so they move around a bit. But they are going to go hang out in a secondary lymphoid tissue, so all those lymph nodes, mucose-associated lymphatic tissue. We've got a lot of lymphocytes in the spleen. And what's going to happen there is they're going to meet their antigen if we've been infected with something that they are designed to fight. They're going to get activated, and then they're going to make their clone army. So all that magic happens in lymphatic tissue. Oh, something about your book. I did want to correct something. All right. So when we were talking about MHC class 1, everybody has one. And they were saying that phagocytic cells don't have one. They actually do. Everyone has one except red blood cells. So MHC class 1 is how am I doing? That's the how am I doing barcode. And then macrophages, dendritic cells, and B cells, these are our antigen-presenting cells, so they have that special antigen-presenting barcode. And I just wanted to make sure to correct that because that was wrong in the textbook. And then the functions of the T lymphocytes, We're going to keep it simple. So cytotoxic T cells, they target our infected cells and they're going to kill. The T helper cells, I'm just going to group them together, but you do have generals that are special for whatever type of infection that you have. And so like TH1 or T helper 1 cells, they are really good when we have a viral infection. Okay, so they just talk more to the cytotoxic T cells and natural killers. T helper 2 cells, they're better at talking to B cells. Okay, but we're just going to go T helper cell. So Xan, I'm just saying, but you have a whole lot of T helper cells. They're all different, but we're keeping it simple. And then T regulatory cells, these used to be called T suppressor cells. These are the things that turn off our immune system. So once we respond to something, they're going to downgrade it, if you will, and they're also the ones responsible for self-tolerance and prevention of autoimmune disease. Keep it nice and simple. All right, so all lymphocytes, like I said, they're targeted to one antigenic determinant or epitope. So think of them, yep, like assassins. Your immune response is going to be tailored to whatever infection you have. Okay, so that turning it on or finding the right lymphocyte, the first step is going to be antigen encounter. Because if you haven't been exposed, there's no reason to get your assassin excited, right? All right, so half the story. So step 1, we have to have, because we're talking T lymphocytes, they need antigen presentation. So dendritic cells, macrophages, B cells will present that antigen to the T cell. The general has to see it and go, yep, I saw it. And then we're going to get clonal expansion. So chest, the T cell that matches that infection, so that's the selection, is going to expand. So it's going to make a clone army. Okay, and we get this for T lymphocytes that are helpers and T lymphocytes that are cytotoxic. So then we get an effector population. So some of them are going to fight that infection right then. So T helpers talk, cytotoxic T cells kill. And then both of these populations are going to leave memory cells behind for the next time. We're going to watch some videos. We're going to look at some pictures. This is the first time I'm telling the story. All right, let's watch it.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows 2 paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
That's a good one to watch over and over. It's the whole story. Well,
Activate.
there we go. All right. So first step has to be the antigen presenting cell has to find it. And so we're looking at right here. So through phagocytosis, dendritic cells, macrophages, they're going to eat it, digest it, and then they display it in context of the MHC class 2. So antigen presentation is necessary for the T lymphocytes for them to get activated. So it's presented to our T helper cell and to the cytotoxic T cell. So remember, the T helper cell is also called a CD4 cell. And once we have presentation to the CD4 cell, that's when it starts talking. Okay, remember, its job is to talk. So it communicates, coordinates, and sends the confirmation signal. It's a talker. Oh yeah, there we go. And it's going to talk back to the macrophage as well. And interleukin, so between white blood cells, it's also going to talk to our cytotoxic T cells. So cytotoxic T cells, they see the antigen, they get a confirmation signal from the general, and their job is to kill. Okay, so they are assassins. So CD8 cells, cytotoxic T cells, I'm so sorry, they have so many names. And like I said, their job, the effectors, are going to be the cytotoxic T cells. They recognize our self cells that are sick. Okay, so virally infected or cancerous cells. Once we have activation, that is when they are going to clone themselves. Okay, they're going to make an army because one little cell isn't enough to, go beat a bunch of bacteria or virally infected cells or anything else for that matter. So they are going to make a group of them that are called effectors. So that group is going to fight that infection. Okay, and so in the case of a cytotoxic T cell, they're going to go kill. In the case of a T helper cell, they're just going to start talking. They're going to release cytokines. They're going to coordinate. They're going to direct. Another population is going to stay behind and not do anything. They're going to go, all right, we'll be here for the next time. And so they're called memory T cells. Memory T cells can live for decades. The effector cells, they are more of a short-lived cell. So memory cells hang back. They're going to fight the next time you're exposed to the same antigen. So now your response is going to be much faster and much greater. If you are exposed to the same pathogen, which we often are, then you're going to go through the whole thing again, and you're going to fight that infection with the effectors, and then you're going to leave behind even more memory cells for the next time. And so this is how that your immune system adapts and gets better as you age in adulthood. To be honest, you've been in contact with several things this morning already, and here you are in your state of health. Okay, so every time you see the same thing, you get the selection, it meets its antigen, then you get that clonal expansion, we're going to make a clone army, and then they're going to differentiate into those two groups. Part of them are going to go fight that infection, the other part are going to stay behind for the next time. So memory cells are what gives you immunity on subsequent exposure or the next time you're exposed to the same thing. All right, so T helpers read the antigen presentation and they secrete their cytokines. The MHC class 1, so now you have an activated cytotoxic T cell, they're reading that MHC class 1. And this is kind of why a antigen presenting cell has to present it in a different barcode, right? Because if it just only had the 1, then the immune system would think, oh, you're infected. See what I mean? So antigen presenting cells do need us because it's a different message. Like, whoa, don't kill me. Don't kill me. I just found it. I'm just telling you. I'm just here to activate you. All right, one more time. All right, T cells, where are they made? bone marrow first, and then they go to the thymus to mature. Absolutely. Then they leave the thymus. They are either going to be a T helper cell, a cytotoxic T cell, so they get their job, but they've never seen battle before, so they're naive. Then they meet their antigen, which has to be presented to them on an antigen-presenting cell. Who are antigen-presenting cells? Give an example. Dendritic cell. What's another one? Macrophage. And I know I haven't talked about B cells yet, but they're also another one. Okay, so they have to be presented. They can't just bump into it and go, oh, I recognize you. Have to be presented. All right, so now they've seen it. To get an immune response, we also need that T helper cell. We need that confirmation signal. But once we have activation, we get proliferation. So that's a fancy word for making a big clone army. And then they're going to differentiate into our two populations. One population is going to be an effector lymphocyte, which means they're going to fight that infection at that moment. The other population are memory lymphocytes, and like I said, they can live for decades, so they'll be ready the next time there's the same challenge, so the same pathogen, the same disease. The effectors in cell-mediated immunity, we have our cytotoxic T cells. They're going to kill by cell-to-cell contact. We're going to just watch a video in just a second. They kill our self cells gone bad, our corrupt cells. They do it through apoptosis. Our helper T cells, we said, are the talkers. Yep, they're going to coordinate and orchestrate our immune defense, both cell-mediated and humoral, and they also talk to the macrophages, and they also talk to the natural killer cells. They do a lot of talking. Then regulatory T cells, which we're not going to talk a whole lot about, they are a subset of T helper cells, and they regulate the immune system. They used to be called T suppressor cells. They changed the name. So they're going to control our immune response, and they prevent autoimmunity, and so they make sure that we have tolerance to things that aren't going to hurt us. So self-antigens, and also you have your microbiome that's there. All right, so yes, I have a lot of favorite videos. So great voice, but this is how both cytotoxic T cells and natural killer cells kill. Well, that didn't work, did it? There we go.
Inside all of us lurks a serial killer. A killer whose primary function is to kill and then kill again. These are cytotoxic T cells, a specialized member of our white blood cells. They patrol our bodies, identifying and destroying virally infected and cancer cells, and they do so with remarkable precision and efficiency. There are about 5 million T cells in a teaspoon of our blood, engaged in a ferocious and unrelenting battle to keep us healthy. These amorphous blobs move around quite rapidly, pushing out their leading edge and probing their environment as they go. When a cytotoxic T cell finds a cancer cell, membrane protrusions rapidly explore the surface of the cell, checking for the telltale signs of cancer. They kill their targets using poisonous proteins visible here in red. These cytotoxic granules move down special pathways in the cell called microtubules to the interface between the T cell and the cancer cell. The T cell punctures the surface of the cancer cell and delivers its deadly cytotoxins. This is very important in our bodies, where cells are packed together, as it focuses the lethal hit on the target and minimizes collateral damage to the neighboring healthy cells. The fate of the cancer cell is sealed. The T cell then moves on, hungry to find another victim.
Such a great voice, right? Did you feel it? All right, so natural killer cells, cytotoxic T cells, they crawl along our cells, they look for infected self cells. They will then use perforin, which pokes the hole, so like your perforated paper has little holes in it. They shoot it full of granzyme, and then the cell will go through programmed cell death, which is called apoptosis. So it's death without inflammation.